Good morning. It's October 23rd, and this is your daily brief in science. Here's everything you need to know. A recent study reveals the lasting impacts of the COVID-19 pandemic on global health, particularly emphasizing a troubling rise in autoimmune disorders. Research indicates that even mild cases of COVID-19 can lead to cognitive impairments, including brain fog and a decrease in IQ. Individuals over the age of 60 have shown consistently elevated levels of anti-nuclear antibodies, both prior to and following the pandemic. Additionally, the pandemic has resulted in increased mortality rates among younger populations, a group that typically experiences lower death rates. While lockdown measures were effective in curbing virus transmission, they also adversely affected essential behaviors necessary for socioeconomic development. The Facility-Based Addictions Treatment Program has noted a spike in applications for inpatient treatment, reflecting a heightened awareness of recovery options. On a different note, many workers have reported that remote work has improved their productivity, citing fewer distractions compared to traditional office settings. The surge in remote job opportunities across sectors like project management and IT services is also bolstering the digital nomad movement. However, the disruptions caused by the pandemic are hindering businesses' efforts to meet sustainability goals, such as reducing carbon emissions and minimizing waste. Experts suggest that to navigate these challenges, it is crucial to establish clear boundaries and take regular breaks to support mental well-being. In property investment, Kenya has emerged as a notable market, particularly in warehousing for the agricultural sector in Côte d'Ivoire and Ghana. Furthermore, the recent outages of cloud service providers highlight the risks associated with relying on a single cloud infrastructure. Taysha Gene Therapies is making strides in the development of AAV-based gene therapies targeting severe monogenic central nervous system diseases. The company's management team brings extensive expertise in gene therapy development, which is crucial for their progress in this field. Recent studies have shown that intrathecal administration is an effective and minimally invasive method for delivering these therapies, aligning with Taysha's clinical development strategy. The upcoming European Society of Gene and Cell Therapy Annual Congress in Rome will showcase significant advancements in gene therapy research from various organizations, including Taysha, Genethon, and Vivid Therapeutics. Genethon, a leader in gene therapy focused on rare diseases, will present work from 20 researchers, highlighting their commitment to innovative treatments that transform patient care. Notably, Serge Braun will discuss one-year data from a Phase 1-2 clinical trial involving an AAV8 vector-delivered microdystrophin gene therapy for Duchenne muscular dystrophy. Affinia Therapeutics is also set to present new preclinical data on their gene therapy programs for BAG3, dilated cardiomyopathy, and MIBPC3 hypertrophic cardiomyopathy. Their second-generation CNS capsids utilize novel blood-brain barrier receptors, improving the delivery of gene therapies to neurological tissues. Additionally, Louisa Jaws will share findings on a gene therapy that corrected metabolic issues in a mouse model of type 3 glycogenosis, demonstrating long-term benefits. Another presentation will highlight the use of S/MR containing AAV vectors, which can enhance therapeutic effects in mouse models with high hepatocyte proliferation. Affinia's proprietary plasmid design reportedly increases AAV yield by over tenfold compared to traditional methods, aiding in cost-effective manufacturing. Despite these advancements, scientists continue to face challenges regarding the safety and manufacturability of scaling adeno-associated viral gene therapies. This Congress will provide a vital platform for sharing groundbreaking research that could significantly influence the treatment of genetic diseases, including Rett syndrome, which affects thousands of patients. A groundbreaking study from New York University is reshaping our understanding of gene regulation and expression. Led by Professor Alexandra Zidovska, researchers have uncovered critical connections between gene activity, genome packing, and genome-wide motions. Their findings, published in the journal Nature Communications, highlight how transcription-driven motions of genes are essential for understanding human health and diseases, including cancer and cardiovascular disorders. Employing advanced techniques like displacement correlation spectroscopy and high-resolution live cell microscopy, the team was able to track gene motion and chromatin dynamics in real time. By utilizing CRISPR technology, they labeled single genes, enabling visualization of gene dynamics in live human cells. The study specifically focused on the transcription of genes MUCA4 and IL6, examining how their activation contributes to large-scale chromatin dynamics. Transcriptional activation of these genes was confirmed through inflammatory stimuli, with nascent RNA visualized to demonstrate active transcription. The researchers theorized that molecular motors powered by ATP drive the movement of DNA within the cell nucleus, although further investigation is needed to fully elucidate these mechanisms. Zidovska emphasized the importance of understanding these transcription-driven motions for comprehending the organization of the human genome, which contains 2 meters of DNA compacted within a nucleus just 10 micrometers in diameter. The spatial and temporal organization of the genome is crucial for cellular processes and has significant implications for the central dogma of biology. This research not only enhances our understanding of the physics of active living systems, but also provides insights into the behavior of the human genome. The study revealed that in low compaction regions, active genes drive genome motion, whereas in high compaction areas, the compactness of the genome influences gene motion. Analysis showed that both MUC4 and IL6 exhibit subdiffusive motion patterns, with their dynamics changing upon activation. A groundbreaking study has emerged from the International ENIGMA Consortium, revealing significant connections between genetic variants and brain disorders. Conducted by a team of 189 scientists and involving over 1,000 research labs across 45 countries, this extensive research was partially funded by the National Institutes of Health. The study analyzed DNA and MR1 brain scans from nearly 75,000 participants, focusing on key brain regions such as the hippocampus, amygdala, and thalamus, which are crucial for memory, emotion regulation, and movement. Researchers identified 254 genetic variants that influence the volume of these brain regions, shedding light on their roles in disorders such as ADHD and Parkinson's disease. Through genome-wide association studies, the research explained up to 10% of the variance in brain volume among participants, providing new insights into the relationship between genetics and brain structure. However, it is important to note that the study is correlational, necessitating further research to establish causal links between the identified genetic variants and the associated disorders. Miguel Renteria, Ph.D., emphasized the significance of these findings for understanding the biological basis of conditions like ADHD and Parkinson's disease. The research not only links genetic variations to increased risks for these conditions, but also paves the way for potential interventions. This study, published in the journal Nature Genetics, marks a significant contribution to the ENIGMA Consortium's ongoing efforts to uncover genetic variations that affect brain structure and function. As highlighted by Thompson, this research provides a roadmap for future interventions in brain-related disorders, underscoring its importance in the scientific community. Graphene, a revolutionary material, is celebrating its 20th anniversary on October 22, 2024. This milestone marks a significant moment in the world of science, as graphene has transformed research and applications across various fields. Graphene is the world's first two-dimensional material, consisting of a single layer of carbon atoms arranged in a hexagonal lattice. This unique structure makes it exceptionally thin yet incredibly strong, boasting properties that surpass traditional materials. It is stronger than steel and more conductive than copper. The journey of graphene began in 2004 when Professors Sir André Geim and Sir Kostya Novoselov isolated it at the University of Manchester using a simple method involving sticky tape. Their groundbreaking work earned them the Nobel Prize in Physics in 2010, emphasizing the importance of this discovery. The original research paper published by Novoselov and his team has influenced countless studies in material science and nanotechnology. Despite the initial excitement surrounding graphene, the challenges of large-scale manufacturing have limited its widespread adoption. However, researchers like Professor James Baker are optimistic, believing that the field is approaching a tipping point for effective utilization of graphene. Even a minor addition of just 0.1% graphene to materials such as rubber or concrete can significantly enhance their properties, improving strength and durability. In construction, Graphene's capacity to enhance concrete strength not only improves durability, but also contributes to reducing carbon dioxide emissions by lowering the demand for cement. Additionally, the biomedical sector is exploring graphene for applications like targeted drug delivery and advanced energy storage solutions for renewable energy. The Graphene Engineering Innovation Center is working to translate graphene research into commercial products, aiming to bridge the gap between innovation and market application. This has been your daily brief in science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. Tune in tomorrow. We'll be back with everything you need to know.